So now your talk is open for discussion. So who want to ask the questions?
I wonder, do they have like explicit social behavior changes?
Well, so we are watching them. We want to know whether they eventually get the idea of empathy and theory of mind. Because one test is that if you look at a monkey and you say look on the back of the monkey and see whether the monkey understands what you are looking, the monkey will turn the head and see what you are looking. So the monkey will know what you are thinking. And that type of awareness is the theory of mind. People think chimps has that, but monkey no. So maybe having the self-awareness is the first step to theory of mind, or self and non-self, the idea of non-self. Empathy, right? Whether there increased empathy. Well, there are other mammals that also have behavior that we can classify as empathy, like a mouse can help the other mouse to do some behavior that seems to be they have empathy or sympathy with other mouse. There has been this type of studies, but in mammals, I think, or in primate, whether this training can result in what you're interested in, higher social behaviors, is one of the interesting aspects of this line of work.
Okay, second question. Yeah, Professor Yang.
Yeah, thank you very much, Professor Pu, for an exciting lecture. I have some questions. For the last part of the experiment, have you tried to put the mark instead of the face of the monkey? For example, in the butt of the monkey, would the monkey?
Well, we haven't, because the way we establish the association is that we do the establish association between face and image. Right? That's the reason for that is recognition of face in the past was thought to be the most difficult part for monkey. The monkey can use mirror to search for things. They can understand the mirror and the object using the mirror to, for example, looking at something behind a barrier, using a mirror and see something on the other side. This ability using mirror as instrument has been demonstrated in monkey, but not for self-recognition of face. So we train that only on the face. Now, but once that training is done, the monkey will extrapolate that idea. For example, if you see an image, a shining image on the back of the side, the monkey look at the image in the mirror. It's on the wall. The monkey will return and touch that back on the wall. So they know the concept. The mirror is a reflection of what's on this side. Right? This concept can be established by training.
Thank you. Now I have one more question about your first part of the experiment about the field conditioning.
Right.
And so you can show that the field memory is stored partially in the auditory cortex because of the particular setup of the experiment.
Right.
Now, would it be possible to associate, for example, with the visual or other sensory perception with the shark, and so that the memory can be, the same field memory can be stored in other part of the cortex?
Exactly. Right. So you can do visual as a visual stimulus as a unconditioned stimulus rather than sound. Pair that visual stimulus with a shock. Then they would, whenever there's a light stimulation, visual stimulation, that's the same as the condition, as a conditioned stimulus, then they would behave, they would freeze. Right? It's well established, but we didn't look for that. Right? Mm-hmm. So presumably amygdala actually sends a lot of projections into cortex. Besides auditory cortex, there are projection to some other sensory cortex. In monkey, there's also projection to visual cortex. In a monkey, this visual cortex is more important. In mouse, sound and olfaction is more important. So there are a lot of projection from the amygdala, the fear center, to the piriform cortex and auditory cortex, more than to the visual cortex. But now you can do association with any other. The principle, I think, is the same. Right? So you pair the recording stimulus, and somehow the potentiation happens in the projection from the fear center to that region.
So the same kind of memory, so the memory of fear can be stored in different part of the brain.
To be associated with a different signal that's relevant for fear.
Okay, thanks.
Yeah, please.
I really appreciate the behavioral design. That's very beautiful. My question is come to the two photon imaging study to see the pre and post synapse formation. And is it possible that there's already been a synapse there, but it just enlarged, but you could not see before the experiment?
So I think you're right. There are changes in enlargement and shrinkage of the spine. We discount those. We do not analyze those. We analyze the most dramatic change. That's the formation of the entire new structure, the disappearing of the entire structure. The reason for that, we can see actually the dynamics. There are a lot of dynamics going on, especially fine structure of the spine and bouton. We do not count that because that's more subtle and quantitatively more difficult than counting the spine, the new spines, and disappearing of spines. So there's a more subtle change as what you're saying, but we didn't count that. And I'm sure it's going on there. There's, right.
First, thank you for a very beautiful talk. I'm not in the field at all, but I have a question regarding the auditory cortex. So you say, how do you distinguish the ability to hear and the memory? Let me explain my question. Assume you associate the sound with fair conditioning, but rather the mice will run away, not freeze. Now you cut off the feet and repeat the experiment, give the sound. You see the mice will not flee anymore. They stay there. Now, can you conclude the feet is the place for memory?
What we are seeing now, to answer your question, we need to establish the causal relationship between that new formed synapse and the field. Right? So the experiment is, can be done these days by activating those pathways that's being potentiated and see if that pathway activation produces the field. To have a causal relationship, you have to show that pathway is sufficient to create a field. That's the first one. What you're talking about is general wipe out the ability of the animal to
You
do.
cannot hear.
But you have to do specific. If you knock out that particular projection, you do not affect the hearing. The reason is that the hearing goes to thalamus, it still goes to that cell, the cortical cells. It does not necessarily have to affect the projection from the amygdala to that area. Right? So we are potentiating a connection to auditory cells that may help that cell to react better when the stimulus comes in. Right? So in order to say that this is the memory, we have to wipe that connection out. Right? We are preventing the formation of new synapse of that particular pathway. If we eliminate the memory, then we say that's where the memory is. That type of experiment, very specific, right? That particular pathway. If that's where the memory is, we have to show it's necessary and show it's sufficient. What I'm presenting to you so far is a correlation only. There's that change correlates with fear nicely, with a high correlation. But we haven't shown the causality. Right? It's very difficult to show causality in this type of things because it involves so many synapses. So you say that, you say where the memory is stored. The ideal experiment is you know which synapse those memory, which synapse actually change after memory is formed, and you prevent the change of that specific synapse when memory is gone. That's where you need. That's the absolute, the complete experiment. And nobody can do that. Technology doesn't allow you to do that. There are so many synapses. There are billions of synapses there. They are so tiny. It's impossible to find all the synapses that's changed in order to wipe them out to show that they are really the cause for memory. So the causal experiment in memory studies is going to be 100 years before we can get to the final experiment. But if you have enough correlation, now before you have the final causal demonstration, people would say that's good understanding of the system. Right? There are many things in the world you cannot demonstrate a causal relation for sure, especially social phenomena. You will have correlation, but the correlation is a very tight correlation, a lot of correlation. You get more confident that there's a causal relationship in there. Right? That's how this field is going.
Right. So I have a second question. Your argument is that the pattern of synaptic connection change that are associated with memory.
Right.
But how do you know a particular pattern of connection encodes for a particular memory or event?
Right. We don't know. That's exactly the problem I was saying. We cannot find those synapses. We don't know. This is the simplest paradigm. Right? We can say, well, if we have there's a specific group of neurons that's tuned for a particular frequency. Right? If we are doing that, your conditioned stimulus is a particular sound, then if that particular neuron responding to that sound, their synapse gets changed. You get better specificity. Right?
Then what is the codon? Like say in genetics, we know what codon occurs for what.
Right.
But what's the theory in memory? What
Right.
codon encodes?
It's very difficult to relate because in a codon, it's just like you want to see the correlation between gene and a complex disease. It's the same problem. Right? There are so many genes involved, so you cannot get. There's a lot of correlations, but you can. There are risk genes you can identify for a particular disease, but you can never be sure that set of genes give that disease.
Okay, so
Well, more and more technology gets more and more precise understanding of particular synaptic function, right? So we want to know more specifically what memory is stored in what type of synapse, which is a big agenda. Now, how does the neural circuit codes for a particular function? Right? And that's a lot of work in this area. People are trying to be able to identify specific cells that's being activated in a particular memory. If you identify a particular cell, then you're one step better. But that's not enough. You want to know a particular synapse that's activated during particular memory formation. Then you can get to the synapse. But there's no technology available to do that.
okay, let's take a last question from a student. Okay, a student, please.
Thank you, Professor Pu, for your lecture. Just as a general question about LTP and LTD. Does LTP and LTD occur in all brain regions or only in the brain regions that are responsible for experiential learning, actually?
Everywhere we look in the brain, except the neuromuscular junction, has LTP. Everywhere, at one stage, some of the activity disappear in the adult. At some stage of development, every synapse have LTP. LTP is a property of synaptic change that depends on the activity. Right? Now, neuromuscular junction does not have LTP, but we demonstrated if you look at very young neuromuscular junction, before they really fully mature, then there's LTP. Right. So LTP is a property of a synapse that's not fully mature, that sits right in the middle or somewhere in the middle in their synaptic efficiency. They can be tuned up or tuned down. Tuned up is the LTP, tuned down is LTD. That's where the plasticity comes from. In the neuromuscular junction, where we want a very reliable reaction, every time you want to contract the muscle, you have to work. Right? You don't want plasticity there. You want a highly efficient, reliable system. The synapse is highly potentiated to the ceiling, the most mature state. It cannot be further mature. So it can only be depressed, but it can never have LTP in the neuromuscular junction.
Okay, so I think we need to invite Professor Pu to come back again, and more frequently, not 5 years every time. So let's thank Professor Pu again for this.